Hello everyone. Welcome to the low temperature thermochronology online course, which is a very exciting topic in geology. And this course could be useful to many people in different geological topics. This chapter will be presented by Professor Li Ronqi from Chang'an University. So, we are going to talk about chapter 11, which is the thermal parameters of sedimentary organic matter. The content of this chapter in three main parts, which are: what is the sedimentary organic matter, thermal parameters of organic matter, and applications of thermal parameters to study the basin thermal history. And we will talk about each point in detail, and each part will be presented separately. We hope that you can enjoy this course. After you finish this course, you will be able to know the basic knowledge about the thermal parameters of sedimentary organic matter. Now, let's start with part 2, thermal parameters of organic matter. Several parameters are used to estimate the thermal maturity of organic matter, such as vitrinate reflectance RO, Cool rank, CAI color alteration index, Tmax, fluorescence parameter, and biomarkers. So, the most used one here is the vitrinate reflectance, and all these parameters not just can be used to estimate the organic matter thermal maturity, but also to reconstruct the thermal history of sedimentary basin. About the AMICs, which is in the right side, those AMICs under reflected white light. In the top image, we can see the part with the gray color is uniform vitrinate with a little fusinite of the internite group. The second image also contains vitrinite with the gray color in the upper part, and the lower part is containing other types of materials. The lower image is darker than the others because the thermal maturity is higher, and also we can see the fusinite in the lower part of the image. Coal rank is the thermal parameter of the coal geology that measures the thermal maturity of the coal. Here in these tables, we can say that the coal rank is divided in a different degrees from lower to higher degrees with correlated with reflected vitrinite. And here we can see two standards, which are the Chinese and the American ones. The lower coal rank in the Chinese standard is the brown coal, and in the American standard is divided into lignite and subbituminous. The brown coal rank is immature here, and after that it will reach the mature window with high volatile. Volatile here, it means most contains is gas, and with the increasing thermal maturity, here we can see in the image, the gas content will decreases in low volatile until the gas disappear in the overmature window. In the overmature window, and only the cover will be left with no gas. The third parameter of identifying the thermal maturity of organic matter is the color alteration index, or the thermal alteration index. We can estimate the degree of thermal maturity from the color of the material of the kerogen. So the color changes with the increase of the temperature from the light yellow color to the black color. And in this image that you can see, it shows that the thermal maturity of the sporinite is higher than the conedonnates, and the sporinite here is a yellow to dark. With can you estimate that it's in the peak oil generation. For the conedonnates, it's a bright yellow. According to many heating instruments, Eberstein divided the thermal maturity changing according to the color, and it gives numbers related to each color, and this used from the yellow to opacity. So you can see in these charts that the colors are changing with the numbers when the temperature is changing to higher temperatures. And from your work and the clear observation, you can estimate the thermal maturity of the conedonate. Tmax is widely used in thermal maturity of organic matter, and it's the maximum temperature that the organic matter experienced during the experimental work, and the already existing oil in a rock will be released when the temperature reached 300 Celsius. With further increasing of the temperature, a new oil and gas will be generated from the rock until the temperature reach about 55, and you can see in this chart. So after the temperature it reached to 550 to 600 Celsius, the organic matter will turn to over mature. On further heating, we can estimate that the Tmax in the temperature of the maximum of petroleum generation from kerogen by pyrolysis. So the temperature of S2 is the peak oil generation. The higher Tmax is the higher thermal degree of oil and gas materials that experienced. Fluorescence parameter is one of the thermal maturity organic matter parameters, and this work with observing a color under the UV light, and the source of the light here should be UV light. The color here will observe under the reflectance microscope, and in this image we can see the different colors from blue, green, yellow to green, and the bright yellow of organic fluid inclusion. The UV light fluorescence spectroscopy measurement uses a wavelength of 420 to 750, and this wavelength uses to reflect the thermal maturity. So to use the two methods of identifying the thermal maturity, which is from the color and also the spectroscopy, wavelength is a good and effective method. Several parameters like Lammax, Q 65/50 and QF 535 were used for quantitative description, variety of fluorescence colors, and also for determining the thermal degree of kerogen or materials of rocks. So the Lammax here, as you see in the chart to the left part, is the maximum intensity of the fluorescence spectroscopy. Each to the left side of the chapter. And the Q 650 to 500 value is the intensity of at 650 nanometer to the intensity at 500 nanometer, and this could be equal to I 650 over I 500. The QF here, you can see it to the right in the chart, to the right of this slide. It's the ratio of 535 to 750 nanometer flux area to the 400 to 535 flux area. So the length between 500 to 600 for the Q is reach the higher intensity. And in this image that you can see, for these materials, it's reached about the peak oil generation in the brighter color. Fluorescence spectroscopy for different materials and temperatures. In figure A here, when the temperature increases, the intensity of the fluorescence will increase. When in figure B, it shows the shifting wave of the spectroscopy. When the temperature increases, the wavelength or Lambmax will shift to the right, and we call it the redshift. When the temperature is low, the wavelength or Lambmax will shift to the left, and here we call it the blow shift. In figure C, is different spectroscopy in which the wave changes to not the right, which indicates different thermal histories, but still called redshift. Another shift indicated lower thermal maturity in lower temperatures is called the blow shift here, which is a bit to the middle or to the left part. With the increasing of thermal degree of kerogen or material, the fluorescence redshift landmark shift to the right, and the fluorescence color change from the blue, gray, yellow, and red, as you see here in these images. So with the decreasing of thermal degree of kerogen or materials, the fluorescence blow shift Lambmax will shift to the left, and the fluorescence color will change to the opposite side with red, brown, yellow, green to blue. With increasing of thermal degree of kerogen or materials, the fluorescence intensity decrease, and the Q 650 over 500 and the QF value will increase. So in the chart in the right side, we can see that the red line is increasing with intensity because with higher wavelength, so the red line will be weaker. So with low intensity, the wave is high. With high intensity, get into a weak part. Biomarkers of organic matter are like the DNA of the animals, which are a group of organic components, molecules used to identify the maturity of organic matter. The most used biomarkers groups in petroleum geology are the Hobain and the Styrens, and they used to determine the thermal degree. And many experiments give quantitative results of the ratios of different biomarkers with vitrinate reflectance, RO, as you see in this chart. For example, for an immature degree of RO lower than 0.5, the Hubane show different features, and if the thermal degree increases, so the Hubane monochromes will be broken and will disappear. Other Hubane structures in different RO with the transformation ratio will change to higher with increasing of RO, and this is the same as the Stearns group. As you can see, the Stearns group will survive mature degree. The ones here we see in this figure is another group of biomarkers, which is the terebennes. And the terebennes here, the more transformation ratio, the higher thermal maturation, and it has 2 to 3 groups. This is the end of the part 2, and thank you for your attention.